All right, so we're recording. So thanks for coming along, everybody, to this Mur Ligase meeting on the 9th of February. And we have a small agenda, but I'm sure that lots of things will come up for discussion. I was just going to say a few words about what we've been doing here at UCL. And that revolves mainly around starting to think about the synthesis of the AZ compound that we'd love to. So one of the main aims of the project at the moment, which a few of us have been talking about, and Joe's been talking about forever, and Chris has been talking about forever, is the idea that we could maybe get two different Mur ligase enzymes to crystallize with the same small molecule inhibitor. And a great candidate for that was the AZ compound, which was crystallized with Mur-C by the Seattle group recently. And so to get that to happen, we need some sample of the AZ compound. And Joe, I'll ask you in a minute just to say something about how that's going in terms of getting samples from the colleagues at AstraZeneca that we spoke to recently. But just quickly, in case, in the likely event we need to make it, we've been thinking about doing that in our lab, and Yuhang has been trying to develop a decent synthetic plan for that compound so that we can make some and then sort of pass it around. Just see if I can share my screen. It refused to let me do it. Okay, hopefully you can see this now. And hopefully you can see that synthetic schema. So this is a slide that Yuhang made and just goes through the synthesis, which we're planning on doing, which is some lit, some of the original lit and some kind of extrapolated lit. And there's a fairly okay, apart from a couple of early steps which might be interesting, fairly okay synthesis towards the AZ compound, which is that guy. Hopefully you don't see my Zoom controls in the way. That guy there. We, I think, are very easily going to be able to make the separate enantiomers of that, so the positive and then the less active enantiomer of that compound. So Yuhang's just ordering those molecules and is going into the lab this week to get started on trying to get that going. Hopefully not too difficult. This is the bit of the crystal structure that came out from the Seattle group, from Mur-C, and the interesting point about this is that some of the interactions are very clear with the enzyme, between the compound and the enzyme. And the OH, this alcohol group, is sticking out into solvent, essentially. And so that drove the design by Dayang and Yuhang and us over here of this compound, which is exactly the same compound, but with an amine there, which apparently was not made by the original AZ team. And which should retain the same kind of interactions as the original compound, but because of its possession of a primary amine, should do better against gram negatives. To do that requires a little bit more trickiness in the synthesis of getting this diamine compound. And we'll certainly be doing this first with the racemate, just to make sure that we can get the chemistry working before thinking about single enantiomers. So that's the sort of the immediate synthetic plan, is to generate enough of this compound that we can share it around, ideally, from the same batch, so that people can have access to this and see how we go. So that's the first thing I was going to say.
I guess if you make the AZ compound, you can just convert the alcohol to the amine.
Yeah, that's absolutely possible. Yeah, I guess that was going to be plan C. Yeah. I guess it depends a little bit how much we can get our hands on.
Yeah.
Yeah. And then the other thing I was going to say is just an update on the Atomwise situation. So as some of you may know, Atomwise have sent us 92 samples of small molecules that they predicted to be tight binders to Mur-D, I think it was. They're in UCL. We had to modify the contract to allow us to ship those samples to Diamond and to Warwick. And after a lot of really not very much, and a change of personnel in the legal office at UCL, which I wasn't aware of until a few days ago, we look like we might be getting there. We've received the crucial little bit of text from Diamond, and now we're pursuing the same bit of text from Warwick. And as soon as we get that, we can ship the compounds and have those evaluated, which will be tremendous. Okay, so that's, I think, pretty much all. We haven't done any more synthesis since we shipped, the elaborated fragments to Diamond and to Warwick last year. So we haven't done anything else on that. So I think that's where we're up to. Have I forgotten anything, UCLers?
I was just messaging Yuhang because I think Lori had sent us one of those starting materials. She did, right? Yeah. Okay. So we'll just check on that before we start the synthesis and make sure that we don't order it again.
Yeah, great. Thanks. All righty. Joe, did you want to say anything about the interactions with AZ? Because they were really productive, and I'm not quite sure where we're up to on that.
So, yeah, I mean, I think AstraZeneca has been extremely helpful and I would say almost encouraging. I've reached out a couple of times. So basically, what we've done is, so AstraZeneca, just for those who may not know, they have what's called Open Innovation, which is a program where they will share some of their compounds with outside groups. And so obviously that was how initially the Seattle group got a couple of compounds that they did crystallography on against Mur-C, Pseudomonas Mur-C. So we were obviously interested in trying to get those same compounds to possibly, test them against, another isozyme, whether it be E. coli or also against Mur-D. So basically, what I've done was I went back to the original publication, the chemical and biology publication AstraZeneca published on Mur-C, and pulled out 5 compounds. Because one of the things the challenge is with this is that, so AstraZeneca basically, there's and I guess Chris and also Bart Stacker at the Seattle group, in terms of getting things through legal, this is, as Matt has talked about with Atomwise, that once you start getting into agreements, it can become a little bit of a quagmire and can take time. So one of the things we were trying to do was to ask AstraZeneca to share a small set of compounds and hopefully get those via maybe a material transfer agreement. The bottom line is there are 3 compounds that AstraZeneca has available out of that series of Mur-C inhibitors that they have available, they were willing to share. So really where we are right now, I know Chris Dowson has already submitted his proposal, the paperwork and so on. So I'm not sure where that stands, Chris, but, and I'm not sure where the Seattle group is at this point. I mean, the only thing I can say, I don't know if that comes up, but Lori and I had a TC with Bart Stacker at the Seattle group, along with Peter Morinani, who was the actual crystallographer, who's actually stationed at UCB. It's a pharmaceutical company. He was at one point part of kind of the Seattle structural biology group, but now is at UCB, actually here in Cambridge, Mass. And he was very enthusiastic. So I think I'm not sure where the status is with the Seattle group in terms of ordering the compounds on their side, but those things are in progress. And it'll be really, it's a matter of AstraZeneca is willing to share. It's not a question. They have samples for 3 of the compounds. It's a matter of getting it now through the legal process. So it's really now in the hands of the gods, pretty much, in terms of the lawyers. So that's where we are, as far as I know. I don't know, Chris, if you had any feedback, any feedback from AZ on your side in terms of after you submitted the proposal.
No, I haven't, Joe. I'm just looking through my junk mail and all the other lovely places Outlook puts things that I really want to read, and I can't find it anywhere. So I've not heard anything from them. I have already sent one poke directly to the AZ folks outside of the Open Innovations group. So I'm trying to remember who it was. I think it was Kelly Gray. Kelly, was that right? I can't remember. I sent it to, but I copied them in.
So yeah, again, I just have to emphasize that the folks, Kelly Gray especially, is, I guess she's based in Cambridge, UK, and extremely forthcoming and helpful. So So I think we'll get them eventually. It's just going to be a matter of, the timing of that. So we'll see.
But is it essentially with them, and then if the compounds are around, that then it's a discussion between them and Bart, who's just joined. Hi, Bart. Is that the nature of the next discussion? So we don't need to do anything else there. We're kind of just waiting for that to happen.
No, yeah, I assume that there is, after Chris Dowson submitted his open innovation proposal, that there's some process that goes through, and then eventually Warwick will have to sign a document, right? And so it's a matter when AstraZeneca sends the legal document for the, to share the compounds, and then Warwick will have to sign off on it. So I don't know where you are, Bart, if you've actually had any more contact with Kelly Gray at AstraZeneca around submitting an open innovation proposal.
I have not submitted that proposal. She did tell me that I think that's what I have to do. So
yeah, so if you need any help with that, I'm very happy to. I don't know if I can do that almost quote for you, or fill it out and then save it, and you can do the final submission. But anyway, anything I can do to help. It's very. Chris did it. Maybe Chris, you could even make some comment about. It's pretty straightforward, I think, right? I think he did it in probably 10 minutes or something.
No, I didn't actually.
Oh,
it took quite a long time.
It took a long time.
So yeah. There's.
So maybe if you have any pointers to help Bart,
I was just going to go and try and
make it.
See if I could, because you have to add a proposal as well. It's got a clunky thing as well where you can add attachments. You have to add attachments. There's no way of retrieving the wrong attachment. So I had a kind of a whole snowstorm of attachments that went on. I couldn't take them off, so I just left them on there. So perhaps they're still having to read all of them. I don't know.
Well, I apologize for not doing that. I'll try to get on that. Unfortunately, the Seattle Children's. I know many of you guys have worked in companies and stuff with difficult legal teams, but I actually haven't ran across a legal team that's hard to deal with in my own. Except for maybe a Sanofi, I think was pretty bad, but we have a process of escalating signatures that takes about 6 weeks to do almost anything. So, because every person has 2 weeks to do something, and then, anyways, so I should get that started. Have you, all right, Joe, are you in Peter Hirani's speaking?
So I think, just unfortunately, Lauren couldn't make it today. She had another, I think, previously scheduled international conference kind of thing, I think. So Lori's kind of been taking the lead. I mean, I think where things stand, I think Lori contacted you recently. So just for the team, we've actually ordered a set of compounds from enamine based on similar hinge binding chemotype that we looking to the Seattle group to potentially try in crystallography.
Yeah, we have received those compounds.
Yeah, so that's kind of where that's where we are. But I haven't heard anything back from. But you have to send those. I'm not sure how that actually goes. If those end up going over to that island.
Yeah, they have to go to Bainbridge.
Yeah, there's an island. That's where the crystallography facility is. Yeah. And then I guess Peter somehow has support over there to get things into trays and all that. And then he eventually does the collection gets done and he does the analysis all remotely. Is that correct?
Right. So what there is on Bainbridge Island, it's a company that used to be called Emerald Biostructures. I worked out there for a long time as well. But they are the crystallographers there are the structural biology group for our structural genomics project. And they are also the structural biology group now for UCB Pharma, because they were purchased by UCB Pharma. And so it's all sort of the same group and people. And Peter Hirani, who was the one who actually did the Mercy structures, at the time he worked over here on Bainbridge Island, but he's since moved to Boston, so he's now on the East Coast. But the way that they operate is sort of distributed, but they've done it for we've done it for years now, like 10 years, where Peter runs, submits jobs and talks to the people all online, and that's all the crystallization is actually done on Bainbridge Island.
Yeah. The distributed
All right.
world.
All right, that's great. Thank you, guys. I was going to move on to ask for updates from Diamond or Warwick that come up, and then just to make sure that we hear from the new people who joined today, they maybe haven't been to the previous meetings. It'd be great to hear about what they're planning on doing. Chris or Lizbe, did you want to say anything about any new developments along the Mur Ligases, and particularly around evaluating compounds and so on?
I'll just pass over to Becca to see what she has to say, because I've not spoken to her for a week, so I don't know where we are with that.
We're actually pretty close. We've done all of Lizbe's in the initial assays, and we're now testing whether they're actually true inhibitors or inhibiting the coupling reaction in the assays. And we're hoping to get that done by the end of this week. And we're also hoping to get Dana's compounds all into the solutions we need to start running their initial testing as well.
Great, awesome.
It's sounding as though you have some hits. As to what they're hitting is another matter.
Yes, yeah, this problem we need to, because of the high concentration from running of them, we need to double check whether they're true or not at the moment. But we're fingers crossed at the moment, hoping so.
And best percentage inhibition?
Not that high, only about, I think the best is 20, but we're pretty sure that one's inhibiting the coupling reaction. So
by that you mean 20% reduction rather than 20-fold reduction?
Yeah, 20% reduction.
So sorry, Becca, I didn't catch which compounds these are.
They're Lizbe's ones, which were binding to the little side pocket. Not any of the ones in any of the substrate bindings places.
So these are the original fragment sets?
Yeah, this is a SGC side project from one of the fragment sets.
Hard to hear you, Lizbe.
Sorry, what was that, Lizbe? You were quite quiet just now.
Okay. So this is a SGC follow-up on one of the pockets that we saw binding one fragment, and just wanted to see if we can build it out. So we got binding, but we showed the compounds to Dana last week. It's really, really greasy, and we're not really sure it's going to inhibit. We were actually convinced it wasn't going to inhibit, so Becca got us a little bit excited with this now.
Okay, and then the ones that you mailed, Dana, are the ones that you and Fahima made. Great, fantastic.
Can I ask one question? So those fragments, you're mentioning, those are XChem screen fragments, yeah? Okay. Thank you.
All right. That's great. So Gustavo and Laura, did you want to just say hi? Because you've not been along to the meetings before, and some of us may not know you.
Yeah, hello, I'm Laura. I'm working with Chris Dawson. I used to be in the SGC in Oxford, but now moved to Chris's lab. And I'm going to be doing crystallography on the Mur Ligases project. Hopefully, I'm going to start setting up the first Mur E crystal plates this week. So fingers crossed.
Yes, so I am Gustavo Bezerra. I was a senior scientist at the Wyatt Group, a metabolic and rare disease at SSGC. And since January now, I joined Frank's group as a team leader for protein crystallography. Yeah.
Great. Well, nice to meet you both.
Sorry,
okay,
Laura, I didn't catch what compounds you were going to setting up plates for against Mur E.
So this week I'm going to do APO crystals to do fragment soakings or any other compound soaking. So I'm starting with the APO, trying to reproduce Lizbe's conditions.
Which proteins?
Mur E this week, I'm doing Mur E.
From E. coli?
E. coli, yeah.
Okay, great. Any other comments about any of that stuff that anyone wants to raise? I mean, as it says on the the GitHub issue, we've still got these three main strands: atom-wise fragments and the modification or confirmation of the AZ compound and any of its simple derivatives in a target-based way. So these still remain the three options, and I think the main thing remains that we need this some confirmatory evidence of a new hit, or ideally two enzymes with one compound bound. So that remains the scientific target. Chris Swain just pinged me before the meeting and identified an interesting funding source from the MRC in the UK, which I put on the GitHub issue with the link that's shown there, which has it opens soon and the window ends in May. And it looks quite interesting and relevant, unless I've misread it. So thanks for pinging that, Chris. I mean, May is possible, if we got the data we really want to get in the next month or two, then that's doable in terms of a funding opportunity over in the UK, which could of course impact everybody, but it could be something that's good. So please take a look at that if you haven't seen it, because I think it looks quite good for us. Doesn't help raise money for colleagues and friends in the US, but it would allow us to do something, and on a reasonable time scale. I guess, Joe, you and Laurie are still thinking about funding opportunities in the US that might be relevant for you guys, right?
Absolutely.
Yeah.
Yeah, I mean, I think we I guess since this is an open source program and should be open, I think, the feeling is we really have to have two different funding grants, I guess. The UK has to have their own, and I think the US has to have their own. And this is a matter of trying to figure out the best way, which chemical equity, goes into each of the different grant proposals. That's kind of what we got to work through. But obviously we have to have data first, and then we can make that decision. But yeah.
Yeah, I was looking, there are a few international schemes. One is the Human Frontier Science Program, the HFSP scheme. And it's interesting, the eligibility for that requirement is such that the team members who are applying, which is usually 3 or 4 people across different countries, have not worked together previously. And I think this call constitutes working together, I think, unfortunately. So I don't think we can do that scheme. And there are so few others that are genuinely international.
So we've put in our Warwick Welcome Partnership Funding Award. We've had the first reviewers' comments back. I think the maximum score for each item was out of 5, and 80% of the score items were 5 out of 5.
Right.
Two of them were 4 out of 5, and that was from people saying, "What about the IP?" So I had to write again, just to remind them it's an open source project. I wrote to our tech transfer people, because they said, "You need to talk to our tech transfer people." So I've spoken to the tech transfer people, so explaining we need a new modality for discovery. I've looked at the MRC Infection Immunity Partnership Grant. I was on that panel for 4 years. Infection Immunity have never funded a partnership award, sadly. Just not wanting to get our hopes up too high.
Wow.
Because it's never normally deemed broad enough, the different groupings. So this is normally kind of a healthcare interventions science thing, like how do you deliver and develop a new vaccine, and goes all the way through from discovery to delivery, and it got social science, etc. But I'm not saying let's not have a go. I mean, we could have a little explore, phone up the program manager and just have a chat to them.
Yeah, good idea. Yes, perhaps the open source angle would appeal.
It's a big piece of work to a partnership grant, so yeah, but well flagged, well done.
So Chris, on that partnership, the Welcome Partnership Grant, so that sounds exciting. Do they actually have is that like a rolling grant where there's in terms of getting a final answer, or is there an actual date where awards are announced?
it's an internal pot of money, so it's a better than usual probability of funding. So funding announcements will be made within 4 weeks. I have to get the rebuttal back this week. And it's only a small amount of money, but lots of levered, you know, there was £150,000 worth of leverage. So, yeah. And,
no, that's a critical, I mean, what you're funding there to get the HT, you know, these assays up in high throughput format. I mean, that's just fantastic. I mean, that's just going to make that's going to make the absolute difference in this, you know, project moving forward. So that's, thank you for putting that in. This yeah, a great opportunity.
Yeah, so I just need to write a killer reply to them. Just, I don't know what the competition is. I know there's 2 or 3 other spin-out companies that actually are traditional IP route things. So just got to really sell this as making Warwick look good for trying something new.
If you need any help with the wording on the IP thing, just let me know.
Yeah, great. Will do.
Okay, fantastic. Anything else anyone wants to mention? Because we're on the half hour, and I think that we covered everything. Everyone's busy. Yeah. Okay, great. That's fantastic. I'll try and write up some action items and to-do things on the GitHub, but I think everyone's doing the main thing they're going to be doing to advance the project, which is awesome. Yeah, as you said, Joe, it's really great to see some comments can be tested in Warwick. That's really fantastic. Okay, great. Thanks so much for coming along, everybody.
If there's anything, again, if there's anything I can do to help, I've got plenty of time, so okay. I will do whatever I can to help.
Yeah, my last contract was 18 months from start to delivery, so let's do better than that.
Yeah.
All right, thanks all, and see you next time.
See you then. Bye.
Bye-bye.
Guys.